in general, a lot of work in CRISPR-Cas9 possible nefarious effect would be translocations, but for some reason there is very little interest into what could happen when a cell actually does not repair the double-strand break induced by CRISPR-Cas9. And that's what we aim to understand in clinically relevant models like primary human T cells that are edited for therapy. So we saw that if the cells could not repair the double-strand break in these cells that divide extremely fast, what would happen is that we'd have a truncation of the chromosome. And it's exactly what we saw. About 5% of the cells that we treated with CRISPR-Cas9 lost chromosome 14, and they completely lost it using single-cell RNA sequencing. That's how we could tell, because we couldn't see any genes expressed on that chromosome 14. Now, track the target gene, the gene coding for the TCR alpha chain, is coded on chromosome 14 extremely close to the centromere. And because one of the arms of the chromosome does not code for any protein coding genes, what happens when you have a truncation on that chromosome 14 at the triploccus is you have a functional aneuploidy. No chromosome is about to be found. So we try to even push that further. We looked at cells that were treated both for the TCR alpha gene, TCR beta gene, and PD-1 gene, just like we do in clinical trials. And we found that exactly what happened. Chromosome 14 is lost because TRAC is coded extremely close to its centromere. Chromosome 7, where the TCR beta is coded, you have a truncation of all the genes that are far away from the centromere. Now, chromosome 2, PD-1 is coded extremely close to the telomere, so we don't see any effect almost. And it exactly shows that what happens, some of the cells, some of the primary human T cells that are used in clinical trial, lose a part of their chromosome when they are edited with CRISPR-Cas9.